Okay, thanks, Javier. And of course, thanks to all the organizers of this conference. For me, it's a pleasure to be here presenting my work, Genome-wide bisulfite sequencing analysis of cultivated and wild-ripe species reveals epigenome variation in response to aluminum stress. And of course, first I have to talk a little bit about the context of this work and why to study aluminum. Well, aluminum is the third most important common element in the world, and it's very important, especially in acid soil, because in this condition, aluminum becomes its most toxic form, Al3 plus ion. And most of the cereals grown in these acid soils, especially in tropical soils, it's very common to find this kind of acid soils. And you can see in the image, aluminum can cause dramatically problems in plants, especially because of the growth of roots. So, of course, affecting production, that is the most important for agricultural and economical problems. So what to study rice specifically? So, well, rice is an important crop, as you know, because represent the food security for at least the half of the world. And we select Oryza sativa, that is the one of the most important cultivated crops around the world, and is also considered the most aluminum-tolerant crop compared with other cereals like maize, wheat, sorghum, and so on. And why study a wild one species, Oryza glumepatula? It's another rice species, and it's very interesting for us because it's endemic of Latin America, and it's supposed to be adapted to these tropical soils, to these acid soils. So it's interesting because maybe there are several mechanisms, tolerant mechanisms related to aluminum in this kind of soils. And additionally, of course, because we have a big collection of these species, 65 accessions of this genotype distributed along different places in Latin America. So we can compare, for example, tolerant and susceptible species in this wild rice species and find very interesting characteristics. So as you know, because we can hear a lot about this in this conference, plants and humans and organisms can respond and adapt to different conditions, especially environmental conditions, using different strategies, of course, genetic information, signal transduction, modulation of transcription, for example, through transcription factors. But today I want to focus on epigenetics, and especially on DNA methylation. So very briefly, epigenetics is considered DNA and stone modifications which do not affect the DNA sequence and are stable and heritable through generations. So this is very interesting because the changes that occur in the plants in response to any condition, any environmental condition, could be inherited to other generations. And we have this general question, and how are DNA methylation patterns affected by aluminum stress in rice? Are there differences between cultivated and rice wild species? And if there are differences between tolerant and susceptible varieties inside rice species for both cultivated and wild species? So this is like a very general scheme of my experimental design. The most important is here. I have two groups, control and treatment. Of course, treatment is submitted to aluminum stress. Something important is that the pH for both is 4, because I don't want to have into account the pH, the acidity. So I have the same pH for both groups. I have, as I said, sativa, that is the cultivated species, tolerant and susceptible, and glumepatula, also tolerant and susceptible. Then I identified the differential methylated region, that is the unit that I'm going to use to compare between these varieties. And here is a graph that I'm trying to explain how I select these regions along the genome. Basically, I split the genome in windows. For example, this is a window, and I compare the DNA methylation marks between control and stress. So this, for example, is not a DMR because it's similar between both, but this, for example, is a hypo-DMR because it's hypomethylated in the stress condition. Okay, so this is a first image. It's like a first approach. We compare control between wild species and cultivated species. Maybe it's difficult to see, but here we have red and blue lines. And blue is for cultivated species and red is for wild species. And the most important here is that these patterns are very similar between both species. And also, as we see the methylation DNA related with genes and TEs in average, we see that are very similar between both species. Now, when I submit the plants to stress conditions, I found a lot of DMRs, these regions in the genome, and something very interesting is that I'm seeing hypomethylation under stress condition. And this hypomethylation is especially remarkable for this context. When I'm studying DNA methylation, I have 3 different contexts. If you remember, methylation in DNA is methyl groups in cytosines. But I can have cytosines with another G. I can have cytosines with H, that is any nucleotide, and G and C with any 2 nucleotides. And it's very important that this context is different from the other context. And I have a lot of hypomethylation for this context, and especially for the tolerant variety in the wild species. This is very interesting because we are beginning to find something interesting characteristics for this species. Then, where are located these DMRs? Where is happening these changes in the methylation in plants under stress condition? And as you see, the first result that we clearly see is that the patterns are very different. Exist a patterns of variation in the plant. So if you see CG, CHG, and CHH, the main number of DMRs are located in repeats. So in repeat regions in the genome. And something interesting is that for hyper-hypo is quite the same. And also between Azucena and BGI that are the tolerant and the susceptible varieties. So we can see that there is a general variation patterns for these species under controlled stress. But if you see, for example, for genes, the main important changes occurs at CG context. So there are also differences between the sequence contexts where are occurring these changes. For the wild species, it's a little bit more difficult because we have a reference genome, but we don't have all the information about the annotation for repeats, for example. We are working on that. But we can see also that the main number of DMRs occurs in something named other, that is not genes or not promoters regions, but we can say that is maybe repeats and intergenic regions. And we also see a pattern of variation in the genome of these wild species. Finally, I select only the genes that had been affected by these DMR changes. And something interesting is that here, the wild species had a lot of genes that had been, that has this variation of DNA methylation. And here I have a number that is for me very interesting because our genes share between all the varieties and are being, we can say that regulated, but it's better to say that are changing the methylation patterns. So the next steps is to focus on these genes. And to final my talk, I want to tell you something very interesting for me because I found some genes that is being affected for these methylation patterns that had been widely reported as important in aluminum intolerant based on transcriptomic studies. So this is very fast. Some of the methods, some of the mechanisms that Rice used for tolerate aluminum tolerance, and I'm finding 3 of these genes. These are transporters of organic acids that chelate the aluminum outside the cell. And also I have this gene, STAR1, that is transported or UDP glucose, who join to the cell wall, avoiding that aluminum ions join to the cell wall, affecting the plant. And finally, I found this family of genes especially this one, NRAD1, is a transporter of aluminum that introduce the aluminum ions into the cell to be conduced to the back wall and be contrarrested. So this is, of course, just the first results of my work. I'm working in this moment with transcriptomic data to look definitely if these changes affect the transcriptomic, but we can conclude that there is a general hypomethylation landscape under aluminum exposure in rice, that the pattern of distribution of the DMRs along the genome is similar for all the varieties, that there are genes potentially regulated through epigenetics that have been previously related to aluminum tolerance in rice, and as a perspective of my work is, do these changes in methylation patterns affect the gene expression? So I want to thanks, of course, to my team group. This is my institute in my university in Cali, Colombia. This is my group, my supervisor, my students, my co-authors, and this is, of course, my other supervisor who is here, and I want to thanks. And of course, to all the foundation that I have got in to this work. And thanks to you for your attention.
Thank you, Jenny. Very nice talk. Some question. We are a little bit late on time for the next, but any question from the audience or from online? No online questions? Yes, Selene.
Hi, thank you for your talk. So, of course, this would be very interesting to, I mean, the long-term solution would be to incorporate some of these changes into a cultivar, like Oryza sativa. So, how do you envision this? I know it's very early on. But
Yes. Well, this is, of course, very early work. At least it's the first work working in rice and aluminum in DNA methylation. But of course, in our future, we can make several things. For example, having taken into account these changes for recombination, because we know that also affects recombination, and for these crosses between plants is important to know this information. But more important is that, for example, thinking in a very future application, we can use Cas9, for example, for edit specific points of DNA methylation. So if I know that there is a DMR in a promoter that is affecting the expression of a gene that is involved in tolerance, so I can edit this, for example, region, and I can enhance the expression of a gene that is important for aluminum. Of course, this is future application for this work.
Thank you.
Thanks.